In order for an immunotherapy strategy to be effective, you've got to direct the immune system to a target. And in myeloma, we have for many years been struggling with a defined target that will allow you to redirect the immune system towards. And more recently, the B-cell maturation antigen, or BCMA as it's called, was found to be highly expressed on malignant plasma cells by comparison to the normal plasma cell population, which is an important differentiation because you don't want to take out your normal plasma cells because then you induce therapy-related hypogammaglobulinemia. But also it's important that this target is also not expressed on the B-cell lineage as well, because that again gives rise to secondary humoral-immune defects. So BCMA is highly expressed on malignant plasma cells, so it lends itself to become an immune target. And there's quite a lot of biological issues there about heterodimerization, sort of promiscuity of receptors and ligands, which is probably best not to go into at the moment. But let's just concentrate on BCMA as a target. So you can develop a monoclonal antibody that's naked. By that, I mean there is nothing on the end. It will attach to the BCMA, and the naked end will stimulate the natural immune response. It will generate complement fixation potentially. It will generate the phagocytic system to engulf the coated cell. It will engender cytotoxic T lymphocytes to attack and engage. So that's what the naked antibody does. And depending on the subclass of antibody, the monoclonal antibody, depends on how much of each of these domains is the mode of action. But also, and naked BCMA antibodies are in clinical trials development. But perhaps more relevant is to put a payload on the end. So what you do is you attach a cytotoxic that otherwise, if you gave it systemically, may have too many side effects that makes it difficult to tolerate. But if you put it onto an antibody, the antibody draws it to the tumor cell, and then either once it's in the tumor microenvironment, the payload detaches, or the antibody is internalized and it's then detached. So you actually get local cytotoxic delivery. And it's a strategy that's been around for a while. So to come back to the BCMA-directed strategy, Belantamab Mafodotin, or Belamaf for short, is just one of those antibodies. It's directed against BCMA. It will then bring up the Mafodotin moiety, which is the cytotoxic. The antibody complex is internalized, the cytotoxic dissociates, and then delivers intracellular cytotoxicity and killing. And the company who have this as a product have been proceeding through their clinical development program to get Belamaf monotherapy accepted by the regulatory authorities, but at the same time exploring what role adding it to combined therapies, including, backbones such as lenalidomide, dexamethasone, pomalidomide, proteasome inhibitors like ixazomib, etc., etc. And you can couple up with other antibodies. Now, in the UK, Rakesh Popat has been leading a very novel early phase study. It's a platform study. So this is where we have multiple phase 1-2 cohorts where we have the control, if you want to call it that, is Belamaf monotherapy. But then we are opening cohorts where we're adding cyclophosphamide and dexamethasone to this antibody, or ADC, antibody-directed cytotoxicity. So we add cyclophosphamide dex to the ADC, or we're going to add ixazomib, dexamethasone to the ADC, and it's building up, layering up the impact on top of this immunotherapy strategy. That's one way to go. There are other strategies in that nature. So that's an ADC, it's an antibody with a payload. Other ways of attacking the BCMA, everybody knows about them, CAR T cells. And this is where you effectively rewire genetically a T cell so that it will stick to BCMA, because T cells are not naturally generated to attack BCMA, because it's not an antigen, a foreign antigen. So what you do is you give the CAR T cell an artificial T cell receptor that is designed to attack onto, in this case, BCMA. The T cell approximates to the tumour cell, it engages with the BCMA, it becomes activated as a consequence of this man-made T-cell receptor because of the internal signaling moieties on that receptor, and that T-cell becomes activated and kills the tumour cell. And we've seen some really impressive depths of responses with CAR T-cells, most of which, and so far in myeloma, have been directed against BCMA. Problem is, though, that while we are getting real impressive depths of response in advanced disease, I mean, some of these people going into the trials have had like 6 lines of therapy, etc. It's not lasting. There is a measurable PFS. So as a cure in myeloma, unlike diffuse large B-cell, in myeloma, it's not there yet. But it may be that we need to tweak the technology or combine technologies in order to get that. But it's another example of a BCMA-directed immunotherapy strategy. Now, the third class is called T-cell redirection therapy, or bispecific T-cell engagers, or BiTEs for short. And basically what these are man-made molecules that are effectively an antibody directed against BCMA and an antibody directed against a T-cell marker called CD3. And these two antibodies are stuck together, and the different BiTEs, or duobodies, or other names for them, have different chemical compositions, but basically that's what they are. They're two antibodies stuck together, and what they do is they form a bridge between the tumour cell and the T-cell. And as you approximate that T-cell, like you approximate the CAR T-cell to the tumour cell, once they're approximated, they're switched on and they get into killing mode. Now, BiTEs, unlike CAR T-cells, are off the shelf. They're in a bottle. You give them to patients. Initially IV, a lot of the BiTEs are now being investigated to be subcutaneous, but you don't need to manufacture or patient-specific entities, which CAR T-cells currently are, although there's some interesting data about allogeneic CAR T-cells, a topic for another occasion perhaps. But the BiTEs, they can be given, and they can actually stimulate the immune system. The second advantage of BiTEs is that they aren't relying on one T-cell clone that you've generated. They're relying on all the T-cell clones that you can switch on in the person. So there is a potential there. But of course, the big elephant in the room about immunotherapy in myeloma is that the immune system in myeloma is particularly compromised at diagnosis, and with advancing stages of disease and recurring rounds of treatment, that immune system doesn't get any better. So you are looking to, say, CAR T-cells, taking those T-cells out. They're already exhausted. They already will show evidence of immune senescence. They are dysfunctional. You're trying to rewire them, put them back in, and expect them to behave like the T-cell from a normal, healthy person. Similarly, you put a BiTE in there, you're coupling up T-cells that are exhausted and senescent, and you're trying to get them to do a job that they're maybe no longer capable of doing. So CAR T-cells don't last very long, which is why you've got this measurable PFS. With BiTEs, there is a danger that you could drive an already exhausted senescent population to become more exhausted and more senescent. So there are issues about the fundamental tumor immunology going on in myeloma at that point. But those are 4 areas where BCMA can be targeted. Naked monoclonal antibodies, the likes of a daratumumab, although daratumumab doesn't attack BCMA, it attacks CD38. But that's an example of a naked antibody. You can have a payload or an ADC, of which Belantamab or Belantamab Mafodotin is an example. There are CAR T-cells that are rewired T-cells towards BCMA, and ide-cel and cilta-cel are 2 very advanced products that are getting regulatory approval and getting into the clinic. And then you have BiTEs. Now, there were 7 different BiTEs presented as new data at ASH, 5 of which were BCMA-directed. So it's an evolving field in BCMA. One last thing to say about BCMA in terms of a target is that people relapse after they've had BCMA-directed therapy. And why might that be? Well, I've already mentioned immune reasons why. So the T-cells might get more exhausted, may become senescent, might not work. But there is some really interesting work that's been presented, and Nico Munch's group from Boston presented a paper last week before, and Herman Einsler and Leo Raska from Würzburg also published a really nice paper in Nature Medicine that looked at actually myeloma cells losing the expression of BCMA as a potential explanation for developing resistance. Not so much not responding first time around, but losing it as you get clonal evolution, then you're getting biallelic deletions of the BCMA producing gene. And therefore the tumor cells lose BCMA expression. And if they haven't got BCMA on, all of these strategies are not going to work. So we are starting to understand a bit more about the immunophysiology of BCMA in this context going forward.